Hello. So welcome to the session on national genomics and precision medicine infrastructures. So indeed, we can do a lot of science and know in principle how this will help people, but to actually implement this in nationwide medical systems is a whole new, whole different scale of endeavor, and it's kind of similar to the analogy of thinking how to build a train and then actually building the train network throughout the country. Probably precision medicine is even much more complicated than building the railway. So without further delay, I'd like to invite the first speaker, Asa Johansson from Sweden. She will tell about the roadmap of precision medicine in SciLifeLab. Welcome.
Okay, so thank you very much for inviting me and giving the opportunity to talk here today. So there were some very amazing talks this morning, and I hope that I will be able to follow up on this with giving some interesting information here. The last thing we heard before the break was that it's really good to work without any resources at all, as in Estonia. What we are trying to do at SciLifeLab today is a little bit to use and also recruit new resources and make the best use of them for precision medicine. So my name is Asa Johansson, and I'm part of this precision medicine capability, we call it at SciLifeLab, where I spend about 20% of my time. Beside that, I'm a researcher at Uppsala University and also a program director of a master program in precision medicine that just started now in August, and we will have a student-driven session after lunch if anyone is interested to listen to that as well. So I'm going to tell a little bit here today about SciLifeLab. Some of you are probably aware of SciLifeLab and what it is, but I'm going to give you a little bit introduction and how we work to make the best use of the capabilities at SciLifeLab for precision medicine. Okay, so Science for Life Laboratory was initiated in Sweden in 2010. So it's an institute for the advancement of molecular biosciences in Sweden. Initially, it was 2 sites in Uppsala and Stockholm, but since 2021, it has been spread to other places in Sweden as well. So today there are the 2 central nodes in Uppsala and Stockholm still, but also 4 other sites to get more national spread in Sweden. So SciLifeLab consists of 3 pillars. So the first one is the infrastructure. So this is built up to be like a service infrastructure to provide technologies to researchers in Sweden, consisting of about 40 different infrastructure units focusing on different technologies, or some of them are the same as well, but located at different sites. There are divided into 10 different platforms. A total of 500 technology experts are employed and work to help and to serve researchers in Sweden. And we have about 1,400 users every year and are running about 3,000 research projects. This infrastructure is partly funded by the government, by private funders, but also by user fees. So you need to pay for the reagents, but you get, of course, the support by the people that are employed. So it's a big employer in Sweden that actually gives a good career opportunity for many PhDs and master students, etc. So the second and very important part as well is the researchers that are affiliated to SciLifeLab. The researchers are not employed by SciLifeLab, but they're affiliated because they do research that falls under the umbrella of SciLifeLab. It consists today, I really saw these figures are not really updated, actually about 300 different affiliated PIs. And the increase from 200, which is in my slides, is also because the expansion of the different sites outside of Uppsala and Stockholm. And these are the PIs, and of course many people working in a research group are also part of this SciLifeLab community. The most recent pillar to SciLifeLab is what we call the data-driven life science. So this was initiated by the Knut and Alice Wallenberg Foundation, who donated 3.1 billion Swedish crowns, which is about 300 million euros, to start up a program to accelerate data-driven life science in Sweden. And it expects to recruit around 500 data scientists or researchers working with data-driven approaches. So I'm going to give a little bit insight into the infrastructure and the data-driven life science project before I move over to the precision medicine. So I'm really sorry, it's quite small slides here. I'm not sure how much you can see, but so this is just an overview of the infrastructures or the different platforms that we have at SciLifeLab. So we have an infrastructure for genomics that you have heard a lot of earlier today, which is actually divided into the genomics, which focus on any kind of organisms, including humans as well, but also the clinical genomics platforms that is located closer to the clinic. And this I will come back to a bit later. There's a platform for bioinformatics support, for clinical proteomics and immunology, metabolomics, spatial technologies, etc., which are all, of course, very relevant to precision medicine. So the data-driven program, what we call the DDLS program, was initiated then to accelerate the data-driven life science in Sweden and to promote Swedish universities to act at the global frontline and we believe this is really important to give impact on the entire life of the scientists and the society in the future. So the idea here was not really to fund specific technologies or projects, but to recruit competence to Sweden and also, of course, to maintain the competence that we already have. So a big part of the funding goes to positions. So there has been a recruitment of what we call the DDLS fellows, which are for young fellows within 5 years after the PhD, who are then giving a very good startup support to be able to start up the research in Sweden with funding for PhD students and postdocs, etc. There's also funding for PhD students in academia, but also in collaboration with industry and for postdocs. And also, of course, some core staff working with bioinformatics, data support, etc. So the fellow program, if anyone is interested, the second round for applying for this has the deadline on the 15th of October. So if you are within 5 years of your PhD doing data-driven life science with interest in precision medicine, have a quick look at this webpage or just Google DDLS Fellows or SciLifeLab and you will find them, because this is a really amazing opportunity. So this was a little bit about SciLife and what we are. But the idea then with working with precision medicine, because SciLife is of course so much more than precision medicine, came a few years ago with the idea that you maybe should try to build together these different pillars, all the different sites, all the different technologies under the roof of different research area. And to be able to maybe address the major challenges in the society. And at that time, the major challenge was the COVID-19. So the first capability that was started was what's called the Pandemic Laboratory Preparedness, which was actually funded with a lot of extra support from both the government and from the private funders to start up a program to use the technologies and the analytical tools to help during the pandemic and to also build up a capability to be ready if or when there will be a new pandemic. So the second capability to be started up was the precision medicine capability, where I'm one of the co-leads. And the idea here is, again, to try to bridge over all the different infrastructures, all the different sites, bring together the whole community and work together. So a little bit similar, we saw this very nice figure the first thing this morning of this precision PMNET, the network here, is a little bit what we would like to build up in Sweden as well in a similar way. So it's very inspiring to see what you have done here. So our capability consists of 4 persons that have been working most actively in this. So this is Päivi to the left here. I'm not sure if she's in the audience here now, but she's going to present later in the afternoon today about her own research. It's also Eva, who is our coordinator, and Janne. And what we have been doing mainly is to start to draft on this roadmap. How are we going to bring all this together? How are we going to make best use of the capabilities we have for precision medicine? We have also then joined with this data-driven life science initiative, so we now have a co-group, and we have then Janne again, who is the chair for the data-driven life science initiative to focus on precision medicine and diagnostics. So a lot of our focus has been on things that are close to clinic today. We have been discussing a lot with different cancer studies. For example, we heard about the Testbed Sweden earlier today by Bettina, and there are many of these initiatives that are already ongoing where we can just contribute and collaborate with. But of course, precision medicine is so much more. And I think that we heard all the very nice idea with baby-seq and population screening. And indeed, the definition that we have used for precision medicine is in agreement with the health ministers of the EU. That is the characterization of an individual's phenotype and genotype. For example, molecular profiling, medical imaging, lifestyle data for tailoring diagnostics and the right therapeutic strategy for the right person at the right time, and or to determine the predisposition to disease and or deliver timely and targeted prevention. And this is, of course, a very huge field, and we are a little bit standing in between saying that where should we really focus? Should we focus on everything, or in one way we can provide services for everything, but we cannot really work actively in this whole field? So we started to write our roadmap very broad. And I'm sorry, I should have brought this roadmap with me here today, but you can actually Google on just SciLifeLab Precision Medicine Roadmap, and you can find it and download it. We had a paper copy published in June this year. And our overall vision is very general, that Sweden should be a world-leading nation in precision medicine and health. And of course, this is what we share with everyone else in Sweden, and this is what people want to do everywhere. But what we really think that SciLifeLab can do here is that we can accelerate research and implementation of precision medicine through this technology and data-driven approaches. And this includes everything. We did see this very nice slices before, that we have, of course, the environment, we have the lifestyle, and we have genomics, we have other layers of information, but we can also incorporate all of them together and try to make the best out of it. So what we are actively working with is, of course, to promote this technology and data-driven precision medicine at SciLifeLab. And this involves a lot of community building, networking, discussing, bringing things to people together. But also to increase the accessibility to SciLifeLab infrastructure for translational research and clinical studies. And this is a really important point, because what we have done a lot is that we have been serving basic research, even if it includes human samples. There have been, for example, population cohorts and biobank samples that are already existing. But if you really want to move into the clinic and help the patients, we need to be able to analyze the samples as they come into the clinic. So this is quite a big transition with regards to handling samples, but also data flow to create the reports and so on. And some of the technologies like genomics is quite stable. We can quite good do it already today. But moving to some other technologies that are more exploratory, we have a bit more problem with batch effects, etc. So we don't really have the workflow already going on. And of course, we need to find a good way to validate tools that are published in the basic science field and take them forward into the clinic. We also work a lot on enhanced awareness and increase the competence on the technology and data-driven precision medicine. So we are spending a lot of time going to meetings, talk to people, participate in workshops, etc. And organizing workshops is one of the plan, of course, as well. And also to establish an effective partnership between SciLifeLab and key stakeholders in precision medicine, because there's so many people who are interested in precision medicine, and we need to kind of be a little bit flexible with where we position ourselves, because SciLifeLab is not working in healthcare. We are completely academic institution who are not really allowed to give healthcare. That's not our role. So we really need to balance it very well with other people. So we have been trying to do a lot of outreach. For example, we have given them a workshop, try to interact with the clinicians, which is quite challenging because I think this is quite similar in many places, but in Sweden, clinicians doesn't have very much time for research. So they are very busy with taking care of their patients. So it's really hard to get them to come to this type of events to start to discuss how SciLifeLab can help them or contribute to their research. And that is really unfortunate. But of course also then discuss with the more political arena, and here in the middle, the bottom, we have a very nice panel discussion during the Almedalen, which is the big political event in Sweden where we discuss with the health minister, etc., about how to move forward. And there has been, of course, a little bit discussion then on where should we position us, and this is a very nice figure, I think, by Päivi, who did try to illustrate the challenges and these valleys of death in how we go from the bright idea of what we can do with the patients until we're really there in the clinic. And we, with SciLifeLab and the universities, we are today working very much to the left. We having bright ideas that we would like to test. We can publish very good articles, etc. But then we need to move it forward into clinical studies, do the clinical trials, and then ultimately, of course, push it forward into clinical routine for the things that actually works. And these bridges needs to be built, and we need to spend a lot of time there. And I think that the left bridge is actually where we at SciLifeLab play a most important role, and where we can move closer. Today we are on the left side of the bridge, but we can actually start to walk on this bridge and push our results further on. And we have already some successful examples. I mean, genomics, as we heard this morning, is of course the field that people are discussing a lot in precision medicine, and what we are working with already today is of course the patients that come to the clinic that already have some disease or problem. So a few years ago, we established this clinical genomics platform to help the hospital to set up these new technologies. And from the clinical genomics, we also established what is called today the genomic medicine Sweden. And I think that many of you here have heard about genomic medicine Sweden. Which works more on the healthcare side than to try to really implement it into the healthcare. So this is a very, very good collaboration already going on. And what we hope, of course, and also they have overlapping sites, is genomic medicine centers and the clinical genomics centers and SciLifeLab. We all overlap with regards to the locations. And what we would like to do is, of course, to use a bit of this knowledge to also put other technologies into clinical care, because it's really important with the genomics. You can find it already in the babies through these baby-seq examples that we heard before, which is absolutely fantastic. But of course, the environment and the lifestyle will also influence our phenotype. So it will also be very important to have other kinds of technologies that can more or less measure the state where we are today, also measure the environmental factors that influence disease risk. So recent research maybe points a bit more towards that for many common diseases, age-related diseases, maybe the proteomics or the metabolomics that capture the state today might be more important for precision medicine in the future, or as important, I should say. With that, I'd like to thank you for listening, and I'm happy to take questions afterwards during the panel discussion.
Thank you. So let's move on. Next speaker is Eero Punkka from Finland. He's the head of the Helsinki Biobank, and he's going to tell us about how they are dealing with the problem of returning biobank data to the participants. Thanks.
Thank you, and thank you to the organizers for inviting me here. Regards from Helsinki. And I don't know why in the program the title is not as I tried to send, so I'm actually talking about returning FinnGenn genomic risk data to biobank donors. So actually continuing on the theme that we heard this morning from Professor Green. Helsinki Biobank belongs to Helsinki University Hospital, and which is the biggest hospital in Finland with, of course, a lot of patients to be treated. It's actually one of the largest hospitals in Europe also. Helsinki Biobank has relatively large recruitment area. We have 2.3 million people in our collection area, mainly because most of the people are collected near the coastline and the southern coast in Finland. The mission of our biobank, or hospital biobanks in general in Finland, are bridging the research and healthcare. And in this model, which you see here, you see that of course, our primary function is to deliver samples and data to researchers on the right-hand side, and we are getting the samples and the consents and also data from the clinics and also from the researchers' collections. But on the right-hand side, you also see another important task that we recently have encountered, which is actually returning the data back to the patients or the sample donors, and also returning the health-related data to clinics. And this is where I'm focusing now. In the biobank consent, you see here our consent form, and we ask everybody whether the sample donor wants to know about health-related findings. And typically in Finland, maybe 98% tick this box. So people are really interested in knowing what the data means to them. The FinnGenn project started in 2017, and it's just recently started its last 4-year period for analysis. And in this project, the biobanks in Finland have gathered 500,000 samples. And these are all genotyped and then linked with the health data coming from health registers and also hospitals. So the starting point for us was actually this returning data from FinnGenn. And because we know that there is also risk-related genomic data associated with the diseases. And now about 300,000 patients' data has been returned to hospital biobanks in Finland. And we started, just when the COVID started, we started actually a project together with the cancer clinicians and geneticists within the hospital to utilize the genomic data in healthcare setting. We chose BRCA2 and 1 and 2, and also PALB2 risk genes, because we knew that they were kind of okay for everybody. They are actionable, and the risk that they carry for cancer is very high. We started by piloting the process first in Helsinki Biobank, and now we are actually widening it to cover the whole country. And we have collected feedback during each phase, so we have actually asked the sample donors by the time when they receive the first information and 6 months after how they feel. I will also tell you about those results. This is the process we are now following. So first, of course, we look for the pathogenic risk variants from the FinnGenn data that is returned to biobanks. Then we identify the sample donors, because we obviously know the identity of the persons, even though they are pseudonymized in the FinnGenn project. Then we make the internal validation, which is a must especially in with genotype data. So because we have the sample, we take it to the clinical genetics lab and make a clinically valid evaluation of the finding. Then we recontact the sample donor because actually the Biobank law gives us the right to recontact the patient. So we do it, and we ask for a permission to transfer the data to healthcare. So that's an additional consent that we have to collect from the sample donors. And once we get that, the information is transferred to healthcare, where the patient is asked to come in and give another sample to revalidate that, okay, that we haven't, nobody has done anything wrong in the process. And after that, of course, there are, things like genetic counseling and follow-up and care are delivered according to the guidelines. As a Biobank participant or sample donor, this is how she would see it. So we actually send letters. We use the old method of sending letters. About 75% of the people who receive or to whom we have sent the letters actually respond. And they respond by logging to a portal. So we have developed a portal where we explain the background of the findings, and then we ask for the permission to transfer the data to healthcare. And then there's a phone call actually from the Biobank telling, explaining the preliminary results, and then we write a referral to the heredity clinic. After that, the sample donor will be called in to give a new blood sample. And then the genetic counseling takes place. Here are the results from of the opinions of the sample donors. And as explained this morning by Professor Green, people actually like it and they expect it. If you look at the questions here and the answers, they assume that they get information because they have ticked the box, and that's why they have participated in the Biobank activities in the first place. They don't find things to be difficult, and they think they benefit from preserving the genetic information. And they are also happy with the counseling efforts that we are doing. And very importantly, nobody is regretting. So there is nobody who regrets. Of course, there is the 25% who never respond. We don't know what they think. And they also tell us that these samples and specimens we have, they should be more widely used in genetic risk screening. Here are some comments from sample donors, some free comments from them. And reading some of these are really touching actually. So people think that we are actually in the business of saving lives. So we don't have any negative feedback. We were expecting but so far we haven't got any negative feedback. We have also estimated the cost effectiveness, because that's naturally a question that everybody asks. How much more will it cost? But actually we have now, the first analysis which has been made by experts at Eero, which is a company in Finland specializing in health economics. They have shown that actually this saves money. These results will be presented next month in Copenhagen at ISPOR conference, so I'm actually not allowed to reveal anything more, but I'm just showing these green bars to convince that actually it is also cost effective. And we are now extending the analysis also to cover ovarian cancer and cascade testing. So basically the families, to see what impact it has on the cost effectiveness. Okay, so conclusions. These are the results from our pilot study, which we actually run, which we finished last spring in our biobank. There we had 34,000 samples or sample donors. We found that 0.6% of them carried these, at least one of these pathogenic risk genes or gene variants. And we also found, the same thing that Professor Green found, that only 74% of the carriers were unaware of this mutation. So most of the people do not know. They walk there happily and they don't know anything about these risks. And now we are actually extending, as I said in the beginning, we are extending this analysis to all FinnGenn sample donors in hospital biobanks. And what we have shown is that screening these FinnGenn results enriches greatly the probability of identifying the mutation carriers. And we have also shown that the genomic information can be used for identifying individuals with a significant cancer risk. 74% of the carriers had no information. Biobank donors in Finland support the biobank activities. We know it, and that's maybe part of, or the reason is perhaps that the people believe or trust the government in Finland, so that's why they also trust that the things we do are okay. And we have also now shown that this preventive care path model is cost effective, which is important for, of course, for decision makers. And now we are extending this analysis to cover all the 300,000 hospital biobank participants in FinnGenn. And we estimate that by the end of this year, we have contacted 200 individuals based on this process. We are also extending this now to other tier 1 diseases, and the first one is Lynch syndrome, so bowel cancer, that we are studying currently. And that's it. Thank you for listening.
Okay, yeah, thanks. So again, write down your questions. I have written down some so that you can remember and ask them later on. Let's move on and come closer, or actually come here to Latvia. Next speaker is Monta Briviba, the head of the core facility Genome Center at the Latvian Biomedical Research and Study Center. And we shall hear what's going on here in Latvia.
Hello, everyone. At first, I wanted to express my great pleasure to speak here today. I heard some very impressive presentations already, and I feel truly inspired. So, the topic of my presentation is National Genomics Initiatives and Projects in Latvia, and I'll talk about those projects that are implemented in Latvian Biomedical Research and Study Center. And I'll start with a brief introduction back to history. So we have a long-standing experience in genetics and biobanking, and it dates back to 2002, when the Human Genome Research Act was accepted. It is still valid. Nevertheless, another biobanking law is being developed at the moment. Then 2 years later, Latvian Genome Center is established, which still serves as one of the main resources for the infrastructure promoting the genomic research. Then in 2006, National Biobank is being developed. In 2011, Biobank becomes a part of National Research Center. And in 2016, Latvia joins this biobanking research infrastructure and also translational research infrastructure communities. In 2018, Latvia joins 1+ Million Genomes initiative, and there are also important improvements made regarding the sequencing infrastructure in Latvian Biomedical Research and Studies Center. And finally, in 2020, Latvia joins EMBL. And regarding this infrastructure and resources promoting the genomic research in Latvia, I wanted to highlight this core facility, Genome Center, which provides state-of-the-art biobanking and omics technologies. And the service that the Genome Center is providing, it starts with the sample collection and it ends even in next generation sequencing. And you can see that we are very well equipped at the moment. We have several sequencing platforms available, both Illumina and MGI. Recently, we also started to use this MGI platform, DNB-Sec T7, which has significantly increased our sequencing throughput. And then we also have library preparation optimization platforms available, some smaller scale machines such as droplet PCR. We have modern facilities now with pre-processing equipment, and we have recently upgraded also the sample storage equipment with a freezer, which allows us to do the barcode-based sample selection. And together with a modern laboratory information management system, now we are able to do the sample cherry picking much faster than we did it before. And the other part of the Genome Center, which is very important, is the National Biobank, which currently holds the biospecimens together with the associated data from more than 40,000 individuals from Latvia. And the Biobank, it serves as the infrastructure resource for both scientific research and also some international collaborations. For a great proportion of the samples, we do have the genotyping and other types of omics data available and stored at Latvian Biomedical Research and Studies Center. We also ensure the access to National Health Registry data for these biospecimens and individuals. And we serve as the Latvian national node for the BBMRI ERIC as well. And I hope you can see I have summarized the list of either recent or ongoing projects in, that are involving human whole genome sequencing data, that are either generating the data or utilizing already existing data. And you will see that the phenotypes, they are very different, starting from rare diseases to pediatric cancers, metabolic diseases. There's also one, this broad international project, an additional project devoted to development of IT tool to transfer the genomic data in a clinical setting. And I will try to briefly introduce you about a majority of those projects, and I will start with this international one, which is ongoing at the moment in many countries in Europe. So as I mentioned, in 2018, Latvia joined this 1+ Million Genomes initiative, and the project named Genome of Europe is developed under the umbrella of this initiative. And the aim of this project is to establish a world-class European reference data research for research and innovation of healthcare. And we are very proud that Latvia is among those European countries that is actually actively engaged in this project, meaning that we are indeed recruiting individuals and doing the whole genome sequencing at the moment. And this is possible only because we do have a funding for this, and the funding is allocated from the Recovery and Resilience Facility. And what it means at the national scale, it means that we need to involve at least 3,500 individuals in the project. We have already recruited 800 individuals at the moment, and we are running the project only for several months. We are doing the whole genome sequencing with the technology that I mentioned before, this DNB-Sec T7, and the sequencing is done already for 700 of these recruited individuals. And the next plan is to integrate these whole genome sequencing data together with clinical, epidemiological data in a secure data portal. So we are still looking for volunteers, and most probably you will still hear from us about the results of this project. Similarly, the genome of Latvia is also being developed. In other words, it's a development of Latvian genome variation reference in a different project. And these are the preliminary data comprising whole genome sequences from 539 individuals, but we are still continuing the sequencing and still involving new participants. And the preliminary data show nothing surprising regarding the total numbers of identified variants within different variant types. So these are pretty similar to other populations as was expected. But here in the table, you can see the most frequent pathogenic variants. And you can see that some of them are more frequent in our population than they are in, for instance, non-Finnish European population. You can see also the related phenotypes they're listed, such as muscular dystrophy or deafness. And as far as I know, these results are not being discussed with the clinicians yet, but I believe that this will be an interesting topic for future discussions as well. And concurrently, we are also using these data to develop a Latvian imputation reference panel, which is actually already being developed using this set of samples. And we have also tested the performance of this imputation panel. And the imputation panel is used to impute the missing genotypes when we're using the genotyping data. So, and we have already tested this imputation panel by comparing its performance with a 1,000 genomes imputation panel. And what we see here is that our own imputation panel works better in case of common and low frequency variants. And we believe that this worst performance in rare variants is due to the relatively small sample size, which we have at the moment, but we are running the sequencing to increase the sample size. So hopefully this will also be solved soon. And if we're looking at Latvian population, to Latvian population from the global scale, I don't know if the pointer is not very well, but anyway, if we are looking at the global scale and graph B, you can see our whole genome sequences in green, and they cluster together with other European whole genome sequences, which is expected. Then in graph A, you can see the European scale. So there are all the populations presented from Europe, those who have whole genome sequences available. And again, our population is colored in green, and we can see that we do have a distinct cluster here. But the most interesting finding is in a graph C, but I would like to show you it. So, do you hear me? Yeah. So this graph shows nowadays sequences, like data from nowadays or modern genomes. These are colored. And the gray ones, the gray dots, I hope that you can see them, these are ancient genomes, also ancient European genomes. And what we can see here, that we are the dark purple ones here, these are Latvians, and these are the modern data. And we can see that our data actually stand like with the highest degree of Western European hunter-gatherer genetic ancestry, which actually makes us pretty unique from the genomics point of view. And it also justifies the need to indeed include our genomic data into this genome reference of Europe. So then we did this principal component analysis according to the region of origin. And these were regions like Kurzeme, Zemgale, Vidzeme, Latgale. These were self-reported regions. And we can see a notable clustering of genomic data according to the regions represented. And we also see that this clustering actually corresponds to the geographic map, so we can distinguish these geographic regions in genomic data as well. And we see here, again, I would like to show you, so sorry if you will not hear me as well. We see this geographic map, and here is a distinct cluster which is attached to Latvia, and we believe that this could be a missing region, Celia, which geographically is not it's not separated anymore, but it seems like it is still visible or distinguishable in genomic data. And then one way how we can transform these genomic data into polygenic, clinical setting is the use of polygenic risks. And for those who maybe are not aware of what these polygenic risks are, the polygenic risks aggregate the information of common variants, the risk of common variants into one score, which can be then calculated for each individual. And if we calculate these polygenic risk scores for a population or a cohort, then this is the distribution in the picture that you can see that we usually get. And for the majority of people, we will have a low to moderate risk for a specific disease. However, there will be 2 to 3% of population those will be the individuals that are having a higher risk of disease with severe consequences. And we may speculate that if we knew polygenic risks for, let's say, 20 diseases, then we may expect that at least 20% of the population would fall within this high-risk group, and those may be then targeted to either prevent or delay the disease onset. And actually, in the previous presentation, Professor Green mentioned that the percentage is 50%, so it's pretty scary. And we are doing these polygenic risk score calculations for different phenotypes, such as type 2 diabetes, familial hypercholesterolemia. We are also now integrating gut microbiome data together with these polygenic risk scores. But the example that I wanted to share with you today is related again to diabetes, particularly maturity onset diabetes of the young, which is an inherited form of diabetes diagnosed in young people. And it accounts for up to 5% of all diabetes cases. And only for the minority of these patients, we are able to find the causal variants, and the therapy depends on these genes involved. And what we did, we used type 2 diabetes polygenic risk score. We tested 2 models that are available in a PGS catalog. And in the graph, you can see the best performing model. And we used 3 different groups of patients. So in green, you can see there are MODY patients with causal variants proved. No, sorry, those are in blue. So in blue, there are MODY patients with causal variants proved. In green, these are MODY patients that we have not found causal variants for. And in pink, it's a control group. And what we can see in the graph is that those patients that do not have the causal variants found yet, those have actually higher polygenic risk scores than those patients that do have this MODY causal variants found. And the control group stays somewhere in the middle. And this suggests that for those patients that do not have these causal variants, or we have not identified them, the disease could be driven by a combination of multiple common risk variants. And then to continue with polygenic risk scores, we are also participating in the state research program in one particular work package, which is aimed to transfer these polygenic risk scores into a healthcare setting. And this is the first prototype of the polygenic risk score calculator. And we aim to involve there more than 600 phenotypes and 1,500 polygenic risk score models from the PGS catalog. And the idea is that any individual with either genotyping or whole genome sequencing data available could upload his or her data in this portal and get back the polygenic risk score calculated for the disease of interest, but this is still in process. And then I will talk about a few projects that are investigating more individual genomes, and as the most prominent one is this investigation of precision medicine strategies in the treatment of pediatric malignancies, which is conducted in collaboration with Children's Hospital. Since 2019, it is financed by patrons via Mikrotik. And the aim of the project is to improve the diagnostics and treatment quality for pediatric oncology patients in Latvia by developing precision medicine approaches. And there were three objectives. At first, to develop the cohort of pediatric oncology patients in our national biobank, then to perform this large-scale genetic testing, and finally to establish a multidisciplinary tumor board. And we have already recruited 299 patients in our national biobank, and since we know that in Latvia there are on average 50 such patients per year, we can conclude that this cohort is highly representative and valuable. And the large-scale genetic testing is performed for 139 patients, and this covers whole genome sequencing for normal tissue, whole genome sequencing for tumor tissue. Both of these data sets are compared to identify somatic mutations, and we are also running the RNA sequencing on tumor tissue, mostly to identify the fusion genes. Okay, then, I'm sorry, could you please help me with the next slide? Okay, sorry. And the main ongoing activities within the same project. So we are looking for collaborations with more experienced consortia and institutions. We have had already some discussions with, for instance, St. Jude Children's Research Hospital, particularly Professor David Wheeler, who is running the computational work in a similar manner. Then we are struggling with data analysis, particularly with filtering and interpretation, so we are still improving our data analysis pipelines. And we have finally also started to collect stool samples from these patients, and we aim to longitudinally collect these stool samples, at least 6 per each patient, during the therapy to evaluate the dynamics of gut microbiota during the therapy and possibly pinpoint some markers related to therapeutic response and tolerance. We have also done some smaller scale investigations using these data. For instance, we have focused on fusion genes only, and we have noticed a significantly increased number of fusion genes, particularly within the subgroup of acute myeloid leukemia patients in comparing to other hematologic cancers. We have found some previously reported fusion genes, as well as identified some novel ones, potentially targetable ones that needs to be validated. And finally, there are 2 projects in Latvian Biomedical Research and Study Center led by Inna Iņaškina that are covering the field of rare diseases. And the first one is aimed to identify these causative genetic elements in rare disease patients, in those patients for whom these variants have not been identified so far with conventional approaches. And here, 149 patients and family members are already involved, and it covers 40 families and the whole genome sequencing of 130 patients and their relatives have been performed in a form of trios or even more. So very extensive work has been done, and the other project is similar but more focused on neuromuscular disorders, again with 99 patients, 36 families, and whole genome sequencing for 99 patients as well. And there are multiple truly interesting findings there, and as one of examples is here, a family with a family tree there, family with 2 cases of multisystemic mitochondrial disease caused by a mutation in a nuclear gene coding for mitochondrial protein, which was in collaboration with international partners, later also proved in a functional study as reduced mitochondrial respiration. And that's it from my side. Thank you for your attention, and I'll be happy to answer to any questions.
Now let's move on to Estonia. Professor Sander Pajusalu is from both the Tartu University and Tartu University Hospital, and is going to tell us what do we do in Estonia.
Yes, thank you, Mike, and thank you all the organizers for giving me the opportunity to be here and share our experiences. So primarily I'm representing healthcare, so I'm working in a routine healthcare setting, and I'm mostly, at least in my first part, I'm focusing on what we are doing routinely in clinic. But then later on I'm switching gears to more similar topics than we heard from Finland, so how to get research data into clinic. So my main disclosure is that I'm employed by University Hospital, which provides clinical and laboratory genetic services. So where we are at the moment in the clinic, so this is the kind of the things that Professor Green today morning said that he's not going to talk about, because this is like, I think, too boring for those who are in the field, because it's routinely happening in many countries, in many centers. So we are doing pretty well in diagnosing genetic diseases. We are doing this in both pre and postnatal setting. We are already in clinic doing some genetic testing in multifactorial disorders. So what I mean here is those diseases which do not have complete penetrance. For example, some cancer predispositions, but also like genetic thrombophilia or something like that. This is routinely done. Tumor tissue profiling is quite standard, at least for some indications, and pharmacogenomics is also coming to clinic very quickly. And of course, we shouldn't forget about pathogen testing, so at least I think that one of the good things that came out of COVID was that now everyone knows what PCR is. Okay, so in Estonia, the history of clinical genetics is actually really long. So the first so-called small clinic for hereditary diseases was established already in late 60s, and it was called something like Clinical Genetics Consultation Office or Clinic. And then it all started with that, so already a few generations ago. But now in 2022, the hospital of Tartu University reorganized or restructured our clinical genetic services into a clinic, what we call a clinic, and genetics, which is called Genetics and Personalized Medicine Clinic. But mostly when we talk about personalized medicine or precision medicine in Estonia, it's mostly used for in the context of Estonian Biobank and the work from MAIT and his colleagues, and but the other part of personalized medicine, precision medicine in clinic is happening as well. So our Center for Genetics and Personalized Medicine, it's now almost already 100 employees, so and this is the largest service provider for the whole Estonia regarding clinical and laboratory services. So we have 2 outpatient clinics, one in Tartu, the other one in Tallinn, and we are kind of serving the whole country. And the same goes for laboratory genetics, so we get referrals from other hospitals as well, from Tallinn. We have invested a lot in laboratory as well. So now we have our NovaSeq X Plus in-house. We are doing more than 4,000 NGS tests per year in a routine healthcare setting, reimbursed by the National Health Insurance Fund, and all other kinds of tests as well. And yeah, we are really happy about our new toy, which arrived this summer. And now we have the opportunity to do like let's say, whole genomes or whole exomes, everything in one house. So the patients are consulted on the first floor, the DNA is extracted on the second floor, it's sequenced on the second floor, interpreted on the first floor, and the results are given back to the patients in the first floor. So it's on the 2 floors, everything happens in one building, one small center, and it's very, very integrated. So laboratory specialists and clinical specialists are working tightly together. And our sample numbers are increasing. I'm wondering when the plateau will reach us, but we are not seeing that yet at all. So the last bar here is current year until August. So it's 8 months compared to the 12 months on the previous bars. So, but you see that for some analysis and for example also the comprehensive tumor profiling, so this is the last section, we are exceeding the numbers of the last year already, like mid this year. So it's a like 2-fold increase almost. We have used quite a lot what is often referred as clinical exome, so this is this Mendelian panel, but now we are switching that one to whole exomes, and we're trying to use more whole genomes where we used whole exomes before, so for the most complicated clinical rare disease patients. I'm not going too much into details about newborn screening because we have our colleague Dr. Kari Reinson presenting about that tomorrow, so who is interested in our efforts there can go and listen to Dr. Reinson's presentation tomorrow. Okay, so a few words about rare disease research. When I did my postdoc at Yale University in the United States, then they said that Connecticut, so where Yale is located at, is not suitable for rare disease research at all because it's too small. Connecticut has 4 million people, Estonia has 1.3. So what are we doing then? So should we even try to do anything here, or should we even try to find new diseases and do some kind of rare disease work? But we definitely should, and this goes for other neighboring countries as well. Like we heard from the last presentations, then there are very interesting findings in the rare disease world from every center that tries or makes the effort to study them hard. And actually one interesting aspect is the disease epidemiology, which is really hard to estimate in the context of rare diseases in larger countries, because the healthcare is so scattered around different centers. But in Estonia, we are pretty sure that our center sees every and each rare disease, severe pediatric rare disease patient in our clinic. So if we want to do an epidemiological study, let's say how many cases there are regarding, let's say, PMM2 CDG, we just know that. We don't have to study that. So, and this gives us a lot of good insights, and we have done a lot of work in this field, and this has been Professor Katrin Onop's work and her team's work. But now to the other side, or the more common complex disease side and biobanking side. So I kind of feel uncomfortable about presenting this because this is MAIT's work actually, and his colleagues' work, but because he doesn't have a presentation himself, so I think I have to say a few words about that. So Estonian Biobank is a very large biobank considering our population size, so it has more than 20% of adult population. Everyone has some kind of genetic data. They all are genotyped, and many are sequenced, and soon more sequencing data will be available. But this work has been a research effort from the beginning, but now, of course, there is interest, what has been discussed today many times already. So the interest is in returning the genetic data results to patients and making it kind of a healthcare information which can be used. And that's why some national personalized medicine projects have been launched and some have been completed already. And the central question there, which we discussed with the Ministry of Social Affairs, is that how to improve the health of as many people as possible by including genetic data in evidence-based decision-making processes for both treatment and prevention. And the focus here is the prevention and preventive side. And this goes well along with the presentation from Professor Green earlier this morning. And there was this big project which tried to and built the infrastructure on how to do it, how to transfer biobank data to patients. It was an infrastructure project, so these actual services are not live yet, but very soon there is a hope that, for example, breast cancer precision screening will start population-wide in Estonia. And I'm explaining what we mean by that is that the first service most probably will be something, not something like, but exactly like this. So every and each 40-year-old female in Estonia will get a invitation to be part of precision screening. So usually the mammography screening for breast cancer starts at 50 years of age, but now at 40 years of age, everyone gets an invitation. And if they want to participate, then it will be checked whether their genotyping data is available at the biobank. But if not, then they will get the genotyping data from diagnostic lab. So there are two ways how to enter this precision screening program. Of course, they give their consents and everything, and then the data is pulled either from Estonian Biobank or the diagnostic labs to a central genetic data information system, which is then linked to computation environment, which can handle software-based medical devices, and these devices will give out the result, which is a risk estimation. And the idea or logic there is that if a person has a similar risk at the age of 40 than an average person at the age of 50, then the mammography screening should start at the age of 40 and not 50. So that's how we personalize the screening, so that the high-risk patients get the screening earlier. And then they get the routine screening invitation for mammography from the central registries. Okay, but what are the lessons learned from this project? And the main lesson is that focus needs to be on the whole service model, not only the infrastructure. So only the computation, it's not enough. So you need to think about all the aspects, legislations, counseling, everything. And the other thing, just when someone starts a new project, what I emphasize a lot is that think about research and healthcare data differences straight from the beginning, so that you are not in a situation in 5 or 10 years that, oh, this data may not be suitable for healthcare. And of course, preemptive genetic testing is something which is which has its legal and organizational challenges, because it's very different from this indication-based medicine we are doing currently. And also the legislation of many countries, at least I'm sure about Estonia, but I'm sure that the same goes for other countries as well. The legislation or the healthcare organization is built for indication-based medicine and not for the preventive population-based medicine. And this needs to be changed. So what do we need in order to make personalized genetic medicine happen for everyone? Of course, we need many things, and I have listed a few of them, and I think that this is good news, so there is work for everyone, and many jobs. So, of course, it starts with the bright ideas, as we heard from the SciLifeLab presentation, but the translation to clinic is not so straightforward always. And of course, you need data, you need medical devices, you need clinical guidelines. It's very hard to make something happen in clinic if you don't have clinical guidelines, and you need to have healthcare providers on board. And to facilitate all this, a new project is starting, and it's coordinated again by MAIT, and the Institute of Genomics, and it's called Center for Data Enriched Medicine. It's an EU project under the teaming program, and it's a 30 million project where 15 million comes from European Commission and the other part from Estonian government. And we are trying to build an ecosystem which facilitates the translation of research findings into really clinical care. We are not making those medical devices ourselves, but we are supporting and building the ecosystem for spin-offs and industry to come along and really make the tools for routine use. And of course, this is a very exciting but very new project, so I hope you hear a lot about these efforts during the next years. And what's also unique about this project is that we are joining forces with academia and healthcare here. So we have on board Tartu University, but also Tartu University Hospital. We have Helsinki University, and we have Erasmus Medical Center from Rotterdam, and also Erasmus University. So healthcare and academia working together. I acknowledge my colleagues and funders, and I thank you for the attention. I'm happy to take questions.
Right, so thanks. Now please all speakers join, take these nice chairs. And we shall start with questions from the audience, as I read from my instructions here. So please, I believe we have microphones. Do we? So please, yeah, questions. You're like Estonians, too shy. Okay. Do we have microphones, or I shall run? I guess I'm faster.
Thank you, Chair, so much. Thank you all speakers for your really interesting and remarkable presentations. I'm Anna Rieksni from University of Latvia, and I would like to ask a question to Monta from Biomedical Research and Studies Center. I see that there's a lot that has been done. You also joined this 1 million project initiative, and I was particularly puzzled by this Latvian map of Latvia also that corresponds to genome. So how many people have you sequenced from each area, and how big actually is the difference between these areas in Latvia, and how it relates to diseases?
Oh, I'm sorry, I may not be competent enough because I'm just presenting the results of the study that is not mine. But anyway, the number of individuals was those 539, as far as I know. And these are whole genome sequences, but they were supplemented with the genotyping data that it might be 1,500 in total. So the integration of both genotyping and whole genome sequencing data. Regarding the numbers within each region, sorry, really cannot comment that, but I have not my presentation anymore, but there was this PCA plot. So it looked pretty equally distributed. And finally, what was the last question?
Relation to diseases. Do you expect that in different areas there will be different kind of risks for diseases?
At the moment, I would say no, but we have not done such an investigation yet. I see no reason at the moment why we should expect that, since our geography is very, very small, and you saw that comparing to other countries or populations, our population is heterogeneous regarding ethnicity, but it's comparing to other countries, it's pretty homogeneous, I would say. But there is one interesting finding that we recently observed, that when we were calculating these polygenic risk scores, you know that in Latvia we have a majority of individuals coming from Russians. And we saw that the polygenic risk for specific phenotypes was slightly increased for Russians than comparing to Latvians. We haven't investigated it in more details. Maybe there's some kind of technical bias because, as I said, we are conjugating different types of data, but it is possible that there are specific SNPs that are responsible for these differences. So at the distribution, at the graph which I showed, we get for our population, for some diseases, not like one peak, but two small peaks. And we see that in one peak there are more Russians and well in other more Latvians.
Thank you so much.
Maybe I can comment a bit about this. So actually in Estonia we have observed and proven, at least for some, that we see even Estonia is even smaller than Latvia, but some regional differences in disease prevalence. So for example, PKU, phenylketonuria, it shows regional differences, or even inside Estonia. And the other really interesting topic is those Baltic-specific diseases, which I have discussed a lot with Biviba from Latvia. And so this is also like on a European scale, what is unique for Baltics?
Maybe I'll also comment on this question. So we've done also similar stuff for Estonian population, and the first part of the question was how different are these regions within Latvia? I think this question is very similar for Estonia. Basically, genetic structure is everywhere when you look at it. It is just differentiation goes smaller and smaller the more smaller area you look at. But you will see genetic structure more or less everywhere. So we see the similar geographic structure also in Estonia. But the important thing there is that it's not always what you think it is. It's not always derived from the population splits. In this kind of analysis, like principal component analysis, what you saw here, you could also get this kind of results when you have, let's say, differential histories in population size. And then that has effect on population structure, or something's changed. Yeah, and there was, anyway. Next question, please.
Yeah, Marcus Lay, and actually I'm a clinician from Latvia, and therefore my question will be related to this. And thanks for the presentations, and I was really impressed, and it got my eye that most of the presentations were catching the issue somehow on the transition of the research data to the clinical data. And just to note that mainly the research data are done by research laboratories that are not certified for clinical use, and I think that is what was clearly mentioned in the presentation from Estonia. But this was really interesting in the Finnish presentation. So what I understood, that you do have the initial sequencing, then validation in a clinical lab, and then when the subject is invited for the further consultation, then it's actually the third sequencing done on the same subject. And is it really the case? And maybe also the same for Sweden, because the lab is research facilities. So how you are dealing with this issue?
Yes, so I can confirm that the first data comes from genotyping. So, and as we know, genotype data needs to be validated in order to verify the variance. So we have the genotyping data from FinnGenn, and because we have the sample that the donor has given, we can analyze it in a clinical setting, and I think that was really important here, so that we can actually transfer the data to the healthcare. And then in the healthcare setting, that's more or less their routine work that they are following. So they will take a sample and analyze it. So maybe there are too many steps, and I hope in the future we can also streamline the process.
Okay, yeah. Sorry, yeah, I can also comment from our perspective. So SciLifeLab works more on the preclinical side. So I'm not an expert in telling about this process, but what we do see is that we have a lot of research where actually the researchers are not even interested in taking it further because it's so complicated. So they kind of just publish it and then they move on to the next project. And I think that we need to start already there to try to convince that we need to continue this process, really start with this validation and see what is going to be used in the clinic, because there's a big difference between publishing significant results compared to what will actually be useful in the clinic in the future. And I really need that we need to bridge over these steps to discuss with each other and make this possible for the future.
So, but the data generated in SciLifeLab is also research data, or do you also have the arm which is certified under 17025?
No, it's true, it's research data. So the clinical genomics works together with the clinics to set up new tools and help with the validation process. But it's not really right now SciLifeLab's main topic to do the validation. But we are also planning to kind of involve different consultants that can help with the process in projects where it's relevant.
Maybe one way on this subject, maybe Monta, what's the situation in your collab? Is it all research data, or do you do collaboration with some hospitals so that you can? So what's the distinction between research and medical data?
As far as I know, we are doing pure research. We are not doing any routine, at least. We're not running any validations in a collaboration with hospitals yet.
So the lab is not certified under, I think 17025 is the magical number.
Yeah, we are also now focused on whole genomes, so it's even more complicated to prove those findings.
I had the wrong number. Sander knows that.
Yeah, it's 15189.
Oh, okay,
so
sorry.
the medical ISO.
Okay, yeah, please.
Yeah, thank you, Zanda Danberg, Riksdagens University. Thank you for all presenters. I think it's so interesting to see a different experience in Nordic countries, although we are so close. But experience is really interesting. I have two questions, one to Estonian colleague and one to Monta. But maybe I will start with Monta because that's the same topic. So my question was about those reference genomes. So first of all, are you screening only for single nucleotide variations, or are you checking also for large deletions, duplications, and copy number variations? And what is your experience with returning results to the donors on the actionable genes?
Yes, thank you for the question. We are screening for structural variants as well. We have recently developed a pipeline for that, and the manuscript will be soon, so you will have an opportunity to see the results. And regarding the reporting results, this project does not cover any feedback, and we are very strict regarding this. So we are trying to inform all the volunteers that we cannot give you any results back because I think you know it's pretty complicated, and we do not have rights to do so, and we should develop a great collaboration with clinicians. And I know that everyone is busy now, so during this stage when we need to generate the data pretty fast, we are not. We cannot provide any feedback, unfortunately, but these data are available, and each volunteer who wants, he can request his data back, but these will be, unfortunately, raw data. It is how it is.
Yeah. Okay, thank you. And a question to Estonian colleague. It was very interesting. You started screening or with combining genetic data with the breast cancer mammography screening. So at the moment when you have started it, approximately what is the percentage of these target female populations to whom you already have genetic data and you can use them straightforward?
So thank you. So this is not live yet, so it's still in planning phase. So, but it's they are actually procuring the medical device if there happens to be one on the market. So 20% of the data is available from Biobank, and it's cleared for this kind of use in healthcare setting. And the other 80% then needs to be genotyped from the beginning. Of course, I want to mention that we are doing actively monogenic variant testing for families, but this is indication-based. So if you have a family history or cancer yourself at a young age, then for certain indications we do lots and lots of hereditary cancer sequencing with panels. But yeah, so we have done like, I don't know, more than 10,000 panels or something like that in Estonia.
But that will be based on polygenic risk score.
No, this is monogenic. Yeah, but the screening will be primarily on polygenic, yes.
Yeah, so have you calculated also economical impact, how it would be to put all this target population on the mammography earlier, or do the genetic genotyping of them? Is there any?
Yeah, so this is a very valid question and very important one. So so there was a pilot study which calculated this economically, but the problem here, which is a very much larger topic, is that I don't believe that polygenic risk scores will get very cost effective if you assess them one by one. But you need to take it as a whole, so you have to imagine that the data is there already, and then the calculation is really, really cheap, and you get a lot of different scores very cheap, and then you can get like good cost effectiveness. Yeah. But when you have to genotype everyone, then it's pretty expensive still, even when we talk about genotyping.
Okay, thank you.
That's indeed a very valid point, but what I wanted to address also, but let's take the audience question first, and then.
Yeah, hello. So my name is Inga-Marlina, and I'm the clinical pharmacist in Rigi East University Hospital and the region my hospital, and I'm a PhD student. So I have a question for my Latvian and Swedish colleagues regarding the genome. The Latvian colleague was presenting the data of the Western Latvia, which is the place where I come from, and actually I was just wondering what are the similarities with the Sweden in terms of Östergötland region and Upper Småland region, including Gotland, because we used to be like kind of very linked, and the west coast is much closer to Gotland of Latvia, I mean, than to Riga. So have you been thinking about defining like the similarities of Western Latvia and Sweden, taking into account that the most popular surname in the western part of Latvia is Johansson? So, so that's kind of my curiosity, whether you have thought out to, link some kind of similarities with like Western Latvians and Swedes, like in Gotland and Östergötland and Småland. Thank you.
So I'm not sure, it's a really great question, it's really interesting. I have never ever thought about it actually. I don't really work too much with the population genetics either, but I mean, we have these maps together, and I'm not really sure whether I have seen that there's a very big overlap actually. I don't remind it. We often see like there are like a Finnish component to the northern part of Sweden, etc., but maybe not too much. But maybe we will get some more information from this European Million Genomes project.
Indeed, this is, I wanted to say, an interesting suggestion. Thank you. We didn't notice any signs of overlapping indeed, but as soon as we will increase our sample size and analyze it together with other European populations, that will increase the sample size. Most probably we will see something.
Yeah, maybe just focusing on the opposite side of the Baltic Sea, not taking into account all the Swedish population, because it could be quite diverse.
From the ancient DNA studies point of view, I can tell that and the early Bronze Age population of Baltics extended to Gotland, or was very similar to people living in Gotland at that time. That's my other job as a population geneticist.
Because we have quite linguistic similarities with the Gotland as well, so.
Okay, cool.
Thank you.
All right. So, since I don't see anyone in the queue right now, then maybe let's take one topic from the title of the session. The title of the session includes the word infrastructure. So, and this question is from a very egoistic sort of point of view. We are just discussing these things in Estonia right now, and I want to get a sort of a wider perspective on this topic, so that I can seem more smart while discussing these things in Estonia. So the question is about how should we deal with data infrastructure in the era where the amount of data and the complexity of it is well, skyrocketing. Let's say if we just deal with genetic data, but we know that it's not going to be only genetic data, it's going to be all omics data sets. But let's just restrict to genetic data. Already here we have genotype data, we have full sequence data, which is which is not only quantitatively different, it's also qualitatively different, especially when we take into account the curation, maintenance, and all these issues. So, looking at from two perspectives: the generation, curation of the data, and then utilization of the data in let's say, medical setting. So, with this long introduction, how do you see, and this is all for all of you, how do you see this data infrastructure set up in a sort of country setting? Do you see more individual centers who produce the data, different clinics, different research institutions, keeping and curating the data they generate and donating it to central databases for where the data is utilized for different things? Or do you see that a country should probably erect a genome center that also generates, maintains, curates all data and distributes it for utilization?
Maybe I can comment on it. So we have already in Sweden now a national genomics platform that Genomic Medicine Sweden have been working for quite many years. But the idea is that the clinical sequencing should be processed and stored in that place. However, we also had another side, which is of course the research part, that are not really part of, it's not really distributed the same. And I think optimally it would be fantastic to have a national platform for curation and storing and also open access of all data. But I think that sometimes, that was, I think, the comment by Bettina before, that in some countries we have no previous structure, and then it might be easier to start from a national perspective. But in many countries, like in Sweden, we already have so many different structures, and it's really tricky actually to combine the different structures. Not only the clinical side or the preclinical side, but also the different regions, the different university, the different molecular fields as well. Because what we optimally would like to do is also to combine genomics with other types of omics data, with imaging, with the registry data, with everything, and have everything in one place. And I think it would be fantastic, and I think that is what we want to work towards, but it's a long way, I think, to go.
Yeah, I've sort of understood or heard that indeed in Sweden, for example, it's pretty difficult to share data between the regions of Sweden.
Yeah, it is really tricky. In the genomics field, they have been moving a bit forward, but it's still really hard to get access to clinical data for other studies. And there's no way, I can go to we're also proud of our registers, and they're really fantastic in Sweden, as in many of the other Nordic countries. But I cannot really go to the registers and see what clinical data is available for this person at the different hospitals. And because that is completely impossible, and that link would have been fantastic, of course. So, but I mean, we're working with it. I mean, there are a lot of strong voices actually arguing for changing a lot of rules, but we have so many rules all the time, so it takes so much time and energy to change it in a better way.
Yeah, if I may comment, I think the problem very often is that the legal framework kind of keeps the data in its origin. So if you have a health register data in a hospital, so legally you're responsible for that. So the best way actually to combine the data, I think, is to form large projects like FinnGenn, where you are actually collecting data from all parts, combining, putting them in one place, and making the analysis. That's much easier than trying to connect the dots or changing the law, which is kind of an impossible thing typically. So I would say, but in Finland in general, we are happy to have a limited number of biobanks, for example, and 5 university hospitals. I mean, we are working very closely, and we are also having one, like FinnBB, which is the biobank cooperative, through which you can get data from all of the biobanks. So there are efforts also in other sense.
I think I'd like to challenge one sentence you said, that changing a law is impossible. I think this is something that we will have to do in many cases anyway. Like Sander pointed out earlier, that, for example, the way how cost effectiveness is thought of has to change. I would also challenge you or ask what you think all connected to cost effectiveness, the whole idea of clinical trials, and this was already mentioned today before Professor Robert Green, that clinical trials in the area of personalized medicine, should we rethink how these are done? If the drug is targeted for a small genetically defined group of people, should be tested for the whole population, which is way more expensive?
I would say yes and no. Yes is obvious, of course, like you hinted that we should personalize and do it more precisely, everything also including clinical trials. But still the basic ideas behind clinical trials still hold up in the current world as well. So we have to make or put all our efforts into doing good and not doing harm to patients. And this is universal, and we have to still remember those core values of medicine and humanity. And that's why I think that clinical trials are still kind of the same, but maybe it's a question about design.
Would they be more targeted as similar to the thing that they're testing for is more targeted?
Well, of course, yeah. And there are efforts like the N=1 clinical trials, and even FDA approves them and things like that. But there have been a very bad outcomes also from those trials. And for gene therapies, many people don't discuss them and tell you about them because everyone likes to share success stories, but there are other stories as well.
Mm-hmm. Anybody else want to comment on this subject? Okay, then actually I have a question for Monte. I was very intrigued to hear what you showed in one slide, that you are preparing a platform or developing a platform where you return polygenic scores of how many phenotypes? Was it like?
We plan to include more than 600 phenotypes.
600 phenotypes. So first of all, have you discussed this with the health board? Do you think this is going to be a medical device that needs to be registered as medical device, so you think you can avoid that?
I ask this because we have this situation right now in Estonia where we were launching similar thing with 2 polygenic scores yesterday and are now discussing with health board again. So that's one thing.
Well, I was hoping that my colleagues will be here who are involved in this project, but as far as I know, the conversations are not there yet regarding the official registration. And as you said, 600 phenotypes, it's rough.
It's a lot. It's a lot.
I think the aim is to show that this is doable at first, that to show clinicians and also the society that we can actually use these data in clinics, and hopefully in the future we may also register it.
And then follow up, do you plan there to just give the polygenic score, or you plan to add also environmental lifestyle data to arrive at total risk score, so that, because why I'm asking is that our thinking in developing the portal, the similar portal, was that we want to not just give the polygenic risk score, but to try to affect people to change lifestyle. So for example, for type 2 diabetes, we give the total risk score using genetic and environmental factors, and then show very graphically that if you lose 10 kilos, your risk will go much lower. So the aim is to make people eat less, to put it very bluntly. So do you plan to do something like that, or is it just giving back the polygenic risk score?
At the moment, it's just a polygenic risk score, and we are looking forward to also attract clinicians that could consult further these individuals that are willing to do some kind of interventions.
Mm-hmm. Oh, yes.
Thank you. Robert Green from Boston. So wonderful session for everybody and so exciting. As you heard, I'm an advocate for trying to honor the contributions of the Biobank participants by at least starting in some way to give something back to them or offer them the potential for giving something back. The question that you just spoke to is a really interesting one. If you give back something like I'm at a high cardiovascular polygenic risk, then what happens? And do people take any? And it's almost like a Rorschach test about, whether people are optimistic or cynical about the potential for intervention, self-intervention, medical intervention. You get a whole lot of behavioral scientists who say, oh, nothing changes behavior. This won't change behavior either. Now, we did an entire NIH grant on the impact of direct-to-consumer genetic testing, which, as you know, early on was only polygenic risk scores, primitive polygenic risk scores. And we did find that people reported, self-reported, with all that comes with that, that they did make changes, improvements in their diet and exercise when they received higher scores on their polygenic risk scores. We published this, but we could not disentangle cause and effect. In other words, were they generally improving their lives at that moment, making changes in their lifestyle, and as part of that, ordering direct-to-consumer genetic testing, or was there actually a notion that they ordered direct-to-consumer genetic testing and in response to it made a second? Importantly, there was not a big difference between the self-reported improvement in the people who had average polygenic risk and the subset who had increased polygenic risk. In other words, both of those groups reported slight increases in exercise and diet exactly the same.
Okay, that's interesting, and we were kind of hoping that people with higher risk will sort of act more, because you always compare yourself to your neighbor and you don't want to be worse. That's kind of, okay.
I mean, interestingly, what happened to low risk group? Did they got lazy? I mean, how did they interpret?
We always worry that the low-risk group will be falsely reassured and somehow abandon their surveillance and good habits. We did not see any evidence of that. In fact, we did a separate study with a genetic variant supposedly associated with smoking and smoking cessation. Supposedly associated with the relationship between smoking and cancer. And the question there was, if we told you you had a low risk of cancer with smoking, would you be less likely to try to quit smoking? And so there's a real case, a real case in false reassurance. And again, we did not find evidence for false reassurance. We did not find that anybody. And finally, we did another entire study with pharmacogenomic variation in which we asked, would people, the medical establishment was very worried that if you give pharmacogenomic information directly to people, they'll start changing their own medications without going to their doctors. They'll stop stuff or they'll take extra stuff over the counter. We did not find evidence of that. People received their pharmacogenomic information. Less than 1% reported any self-motivated change in their pharmacological regimen. So I guess the total thing is it may impact less than we hope in the current zeitgeist. But remember, in the future, when this becomes more relevant to people's lives, it may have a whole different psychological impact and may be more salient.
If I can just comment on it, because I agree with many of your points there, but maybe when we have specific risk factors, it could be more important. I mean, if you know that you're extremely high risk of dying from COVID-19, for example, maybe you can be prioritized to get the vaccine earlier, or similar to what you try to do with older people during the pandemics. But I think another point of having a polynomial score, which is maybe not communicating to the people, but is of course what we heard in the morning, to communicate it to the health care, so that when you get to the hospital with some symptoms, they can easier pick up where you're high polygenic risk in this phenotype. And there, I think that is a really big gain.
Okay, so it seems like we're just getting started with this discussion. So it is especially sad that this young man over there has been indicating me for a while already that lunch is waiting. And if we stay for much longer, it won't be waiting for much longer. So with that, thank you, all the speakers. And, that's presents. And see you soon at lunch, and then later on. Thanks, bye.